All right, here we go. As we get started here, a couple of things to keep in mind. You should have printed off the fill-in-the-blank version for yourself. And while I know that you are capable of just fast-forwarding through this video and filling in the blanks, that's not really the point. The point is that you need to learn this stuff. We are going to have a test on it, and so listening and following along is your best option. I will try to keep it to a minimum time-wise. Another thing to keep in mind is that you don't have the option to ask questions as we go, since this is not a live version. So please write down your questions, and we will, you can ask them later. You can text me or email me or use Remind to ask me. So let's get started. You all know about the structure of DNA. We've done that. So what is the point of DNA? Well, DNA is the place where you find the instructions for making proteins. DNA is found in the nucleus. The thing with this is we've known for a long time, you've known for a long time, that DNA contains the genetic information for the organism, but we don't ever really talk about how that information actually gets translated into traits. Because that's the step-by-step process. You've got to have DNA that contains the instructions. That DNA has to be converted into proteins because it's the proteins that determine your traits like hair color and eye color and height and weight. And for plants, flower color and how the leaves are attached to the stem. And for a mosquito, how long its wings are and how long its little pokey thing is, its proboscis. But those proteins, we talked about cell structure before, proteins are made in the ribosomes. Ribosomes are found in the cytoplasm. DNA is found in the nucleus. Ribosomes that make the proteins are found in the cytoplasm. DNA is too big to leave the nucleus. The pores in the nuclear membrane are not big enough for the DNA to leave, so that provides a problem. The information, the instructions are in the nucleus. The factory for making the proteins are in the ribosomes at the cytoplasm. So that's a problem. So the way that when you look at this, you can see the nucleopores. If I can get my, there we go. You can see right here the nucleopores, or the pores in the nuclear membrane. And then the ribosomes are the tiny little reddish blackish dots out here, and the DNA that's found in the nucleus has to stay there. So there has to be something that takes the information from the DNA in the nucleus to the ribosomes in the cytoplasm, and that's what RNA does. DNA and RNA are very similar to each other, but they have some very significant differences. Obviously, based on what I've already said, you know that RNA has to be smaller than DNA. That would be right here where we're talking about DNA is double-stranded, RNA is single-stranded. So RNA is half the width of DNA, which means that it is smaller and can get out of the nucleus. Deoxyribose is the sugar in DNA that forms the nucleotide that's part of the backbone, that pentagon shaped in the structure that we draw as we represent the DNA molecule in ribose, or ribose is the sugar in RNA. The difference between ribose and deoxyribose, D means without, oxy, oxygen, so deoxyribose has one less oxygen than ribose does. In DNA, the four bases are adenine, cytosine, guanine, and thymine. In RNA, uracil replaces DNA, or replaces thymine. So this is, I'm using my mouse pad, so this will be really awful. I should have plugged in a mouse. Isn't this fun watching me try and draw with the mouse pad? So uracil is the substitute for thymine in RNA. DNA is only in the nucleus. RNA can move between the nucleus and the cytoplasm, and the complementary base pair ruling is adenine and thymine and cytosine and guanine in DNA, and since uracil is replacing thymine, it is the uracil that pairs with adenine. All right. There are 3 different types of RNA. We will spend most of our time talking about one in particular. But it doesn't matter what type of RNA you're talking about. All of the RNA is made from a DNA template. A template is a model. It's the thing that you use as your base that you're going to copy from. So messenger RNA will be the RNA that we talk about the most. This is what actually carries the information about the protein structure. The other 2 types of RNA are vital in protein synthesis, but it's messenger RNA or mRNA that actually contains the base sequences for making the protein. Ribosomal RNA, rRNA, we won't really talk about a whole lot, but rRNA is there for actually making the ribosomes. Ribosomes and proteins are what make up a ribosome. And then transfer RNA or tRNA is what helps to bring amino acids to build the protein chain, the peptide chain. And just as an FYI, protein, polypeptide, and amino acid chain are all interchangeable terms. So keep that in mind as we go. I will use all of them just to make sure that you're comfortable with hearing all 3 different types. And we'll talk more about tRNA later when we talk about translation. So just to give you the big picture overview of protein synthesis, there are 2 parts to protein synthesis, transcription and translation. In transcription, the goal is to take the information that's found in the DNA and copy it into messenger RNA. And that'll be the first thing that we talk about. The messenger RNA then leaves the nucleus and goes to the ribosomes where, again, it's in the cytoplasm, and that's where translation occurs. Translation is where you use the messenger RNA, the rRNA, and the tRNA to build the protein. You need to have all of those. And I'm sure you can hear Ollie saying hello to you in the background. Somebody must be walking by or there must be a leaf blowing by. Hang on just a second. I'm going to pause for a moment. Okay, hopefully he's done barking. So with translation, you've got to have all 3 of those to keep things moving. So a protein is basically just a string of amino acids that are covalently bonded together. And keep in mind that whenever you're building a molecule, you are going to put molecules together using dehydration synthesis. Don't let the dehydration part of it fool you. That sounds like you're breaking things apart. Synthesis is the thing to key in on. That synthesis means to build something. So when you're looking at a picture of it, here's the nucleus. Here's DNA. Transcription is what happens here, going from DNA to RNA. Then the RNA leaves the nucleus, comes out to the ribosome, where translation occurs, and you take these little purple ball things and you hook them all together into a string of pearls or a string of protein. So in transcription, the main thing to remember is this is happening in the nucleus. It's not happening in. And again, this is when RNA is made from the DNA template. And we don't have to worry too much about this, but just FYI, it's the 3' to 5' strand of DNA that's being used. In other words, 3' is the end that starts with a deoxyribose and it goes towards the end that has the phosphate group. All enzymes, and transcription is an enzyme-driven structure or process, all enzymes have to go from 3' to 5'. We saw that in DNA replication, and it's happening here in transcription as well. For the most part, you're not going to be given the entire DNA molecule, both strands, and be asked to figure out which strand is being used. If you're given a strand of DNA, you can assume that it's the 3' to 5' and just go left to right, because that's the way our brains are taught to read things. The enzyme that's being used in this case is RNA polymerase II. There is an RNA polymerase I. You don't have to worry about what it is that RNA polymerase I does. And each gene on a gene is a section of DNA that codes for a protein. This is going to sound familiar from when we talked about the TRP operon. There's going to be a promoter region, which is the place where transcription starts. This is going to be the place where RNA polymerase II latches on to the DNA. There's going to be a transcription unit, the part of the gene that actually is used to build the protein. And then what you see on here as this little green section, that's the termination region. So the blue part on here is the promoter region. The codons, which we haven't talked about yet, but the bases that are going to be used to actually make the protein are in the red section, and then the green is the termination region. And so in this particular picture, there are 2 genes present. Now, I'm going to stop right here in this video because the program that I'm using only allows 15 minutes per video, and I'm at 12 minutes already. So I don't know if I'll get to the steps in transcription yet today, but you should be checking Edmodo every day, and I will try to make sure that I send out reminders to so that you know when things are on there. I hope that you guys are doing okay. If you guys need anything from me, even if you just need to talk, this is really confusing and unsettling what's happening, and it is for the adults too. Don't let them fool you. But please contact me and we'll figure out how we can be in touch, and hopefully I can offer you some reinforcement, some reassurance, and some encouragement. So there you go, guys. I'll be with you again soon.